Hi New Normal and hello freshman students from the BSCMT. Welcome back to our YouTube channel. In today's video, we're going to discuss about biotechnology and genetically modified organisms. So sit back, listen, and enjoy learning. And these are the learning outcomes for today's discussion. At the end of the lesson, the student should able to first, discuss the application of biotechnology. Second, explain the nature of biotechnology. And lastly, evaluate the implications of genetically modified organisms and potential future impacts. So let us fasten our seatbelt as we discuss the implication of biotechnology and genetically modified organisms.
10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Let us begin by understanding the meaning of biotechnology. Biotechnology is the manipulation of biological organisms to make products that benefit human beings. It contributes to such diverse areas as food production, waste disposal, mining, and medicine. The first achievements of biotechnology were in food production, occurring about 5000 before Christ. Diverse strains of plants or animals were hybridized crossed to produce greater genetic variety. The offspring from these crosses were then selectively bred to produce the greatest number of desirable traits. Repeated cycles of selected breeding produce many present-day food production programs. In the 1960s, an important project used by hybridization followed by the selective breeding to increase food production of quality of wheat and rice crops. Now, let's get to know the modern achievements of biotechnology. Modern achievements of biotechnology include the transfer of specific gene from one organism to another by means of transgenics, the maintenance and growth of genetically uniform plants and animal cell cultures called clones, and lastly, the fusing of different types of cells to produce beneficial medical products such as monoclonal antibiotics, which are designed to attack a specific type of foreign substances. Now let us proceed to the different applications of biotechnology. Biotechnology has been applied in various fields. First is we have the waste management. It is used to create a new biodegradable materials. One such material is made from the lactic acid produced when the bacteria individual lactic acid molecules are joined chemically. Thus, they form a material that has the property of plastic but is biodegradable. Widespread production of plastic from this material is expected in the future. Second is in the mining industry. The bacterium Theobacillus ferrooxidans can use the molecules of copper to form the compound copper sulfate, which can be treated chemically to obtain pure copper. This mining process is used only with low-grade ores. As high-grade deposits are exhausted, the amount of copper using this process will likely increase. Then we also have in the field of medicine. The most dramatic applications in biotechnology are employed. Factor 7 was produced for use by homophiliacs. In this biotechnological procedure, the human gene that codes for the blood clotting protein is transferred to hamster cells grown in tissue culture. Now, let us discuss about genetic engineering, or also known as recombinant DNA technology, as well as its techniques. Genetic engineering or recombinant DNA technology is the alteration of organisms' genetic or hereditary materials to eliminate undesirable characteristics or to produce desirable new ones. It is also used to increase the plant and animals' food production, to help dispose the industrial waste, to diagnose diseases, improve medical treatments, and to produce vaccines and other useful drugs. Included in genetic engineering techniques are the selective breeding of plants and animals, hybridization, or production between different strains or species. And lastly is the recombinant deoxyribonucleic acid or DNA. Genetic technology is the means by which knowledge from genetic studies is applied to medical research, criminal investigations, agriculture, and other areas. Genetic engineering uses the selective breeding and DNA profiling or fingerprinting, and we'll be discussing them one by one. Now, let's get to know the applications of genetic engineering. First is we have selective breeding. It is first known technique still used today. It is done to increase food production. In here, only those plants or animals with desirable characteristics are chosen for further breeding. Corn has been selectively bred for increased kernel size and number and for nutritional content. Through selective breeding, cattle and pigs have become main source of animal food for humans. Dogs and horses have been bred selectively for thousands of years for work and for recreational purposes. Second is the DNA profiling or fingerprinting. This is also termed as DNA profiling, wherein the process itself produces a unique genetic maps or fingerprint for each individual. This technique has been proven to be effective especially for linking criminals to a crime, for freeing innocent individuals, and for settling disputed paternities. To make a genetic fingerprint, researchers isolate the target area of DNA and treat it with a combination of protein called restriction enzymes. These enzymes recognize specific sequence of base on the DNA and split it apart at those sites. Because the target DNA is different from each individual, each enzyme will split the DNA at a different site or may not even split at all. The result is a combination of fragments of different lengths. When these fragments are separated by length on a special piece of paper, they form a pattern that looks like the barcode used for identification products in the supermarket. This is the genetic fingerprint, which may then be matched with another sample of DNA fingerprint. Now let's get to know the uses of DNA profiling. DNA profiling is used in several ways. First, it is used to diagnose inherited disorder in both parental and newborn babies in hospitals around the world. This Disorder may include cystic fibrosis, hemophilia, Huntington's disease, Alzheimer's disease, sickle cell anemia, and many other genetic diseases. Second, it helps predict prospective parents understand the risk of having affected child. Third, by the DNA fingerprints of relatives who have history of some disorder, it is possible to identify DNA patterns associated with the disease in question. Fourth, DNA fingerprint links suspect to biological evidences such as blood or semen stain, hair or clotting found at the scene of the crime. Fifth, DNA fingerprints establish paternity in custody and child support litigation. And lastly, DNA fingerprints help identify casualties or persons missing in action. Now let us move on to gene splicing. Gene splicing is a technique which scientists use to directly alter genetic materials to form recombinant DNA. In gene splicing, one or more genes of an organism are introduced to a second organism. If the second organism incorporates the new DNA into its own genetic material, recombined DNA results. The introduction of new genes into an organism essentially alters the characteristics of organisms by changing its protein makeup. In gene splicing, DNA cannot be transferred directly from its original organism, known as the donor, to the recipient organism, known as the host. Instead, the donor DNA must be cut and pasted or recombined into a compatible fragments of DNA from a vector, an organism that can carry the donor DNA into the host. The host organism serves as a factory where the recombinant DNA can be duplicated in a large quantities. The produced protein can then be removed from the host and used as a genetically engineered product in humans, other animals, plants, bacteria, or viruses. The donor DNA can be introduced directly into an organism such as injection through the cell walls of plants or into the fertilization egg of an animal. Now, let's get familiarized with the applications of gene splicing. The advantages of gene splicing are proved in its applications. First is in the food production. Recombinant DNA is used to combat one of the greatest problems in food production, the destruction of crops by plants, viruses, or insect pests. Scientists also have developed transgenic potato and strawberry plants that are frost resistant, potatoes, corn, tobacco, and cotton that resist attacks by certain insect pests, and soybeans, cotton, cords, and oilseed rape that have increased resistance to certain weed-killing chemicals called herbicides. DNA has also been used to prove or improve crop yield. The transferred gene causes the new plants to produce more grain and a shorter stalk with fewer leaves. The shorter plants also resist damage from winds and rain better than taller varieties. Gene splicing technique also applied to animal food production. The growth hormones genes of rainbow trout was transferred directly into crops' eggs, producing carps or 1/3 larger than the normal size. Other engineered fishes are salmons and trout, which have become more resistant to infection by blood virus. A hormone is also injected into dairy cows, increasing their milk production. Second is pollution control. Genetically altered bacteria can be used to decompose many forms of garbage and to break down petroleum products, and recombinant DNA can be used to monitor the breakdown of pollutants. We also have in the field of medicine. The hormone insulin is cloned in large quantities because it is available only in limited quantities from hogs. Another important genetically engineered drug is interferon, a chemical produced by the body in tiny amounts. Engineered interferon is used to fight viral diseases and as anti-cancer drugs. Another is the recombinant DNA is also used in the production of vaccines against diseases. A vaccine contains a portion of an infectious organism that does not cause severe damage or disease, but does cause the body's immune system to form protective antibodies against that foreign organism. Lastly is cloning. In cloning method, scientists remove the DNA containing nucleus from a female's egg and replace it with a nucleus from an animal in a smaller species. The scientists then place the egg in the uterus of a third animal known as the surrogate mother. The result of the birth of an animal that is newly genetically identical to the animal from which the nucleus was obtained. Such an animal is genetically unrelated to the surrogate mother. Now let us discuss further about genetically modified organisms or GMO, or also known as the genetically engineered organisms or GEO. A genetically modified organism or genetically engineered organism is an organism whose genetic material has been altered using genetic engineered or engineering techniques. These use DNA molecules from different sources which are combined into one molecule to create a new set of genes. This DNA is then transferred into an organism, giving it modified or novel genes. A transgenic organism is a subset of GMO of organisms which have inserted DNA that originated in a different species. Genetic modification involves the insertion or deletion of genes. When genes are inserted, they usually come from a different species, which is a form of horizontal gene transfer. To do this artificially may require attaching the genes to a virus or just physically inserting the extra DNA into the nucleus of the intended host with a very small syringe or with very small particles fired from a gene gun. Now let us discuss the different uses of genetically modified organisms. Genetically modified organisms have widespread application. They are used in biological and medical researches, production of pharmaceutical drugs, and experimental medicine, and agriculture, such as the golden rice. First is one of the transgenic microbes. In the past, bacteria were the first organism to be modified in the laboratory. These organisms are now used in producing large amount of pure human protein for use in medicine. Genetically modification or modified bacteria are used to produce the protein insulin to treat diabetes. Similar bacteria to produce clotting factors to treat hemophilia and human growth hormones to treat dwarfism. Transgenic bacteria can be reduced to formulation of cavities. Transgenic microbes can kill or hinder tumor, facilitate crop growth, and produce chemicals which are toxic to crops past. Second is transgenic animals. Transgenic animals are used as experimental models to perform tasks with genes whose function is unknown. Genetic modification can also produce animals that are susceptible to certain compounds or stresses for testing biomedical researches. One good example of transgenic animal is the transgenic fruit flies. They are used to study the effects of genetic changes on development. Another is transgenic mice to study cellular and tissue. A specific response to disease that cannot be studied in mice. The use of transgenic fruit flies. And then we also have transgenic fish. Is created to increase the spread of development and reduce fishing pressure on wild stocks. And lastly is the transgenic plants. Transgenic plants are engineered to possess several desirable traits including resistance to pests, herbicides, or harsh environmental conditions, improve product shelf life, and increase nutritional values. For instance, the Bt maize is a corn genetically modified in order to sicken or kill insects that try to consume it. Specifically bred varieties of lettuce can be grown in water containing dissolved nutrients rather than in soil. The safety of GMO in the food chain has been questioned, with the possibilities that they could introduce new allergens in foods or contribute to the spread of antibiotics resistance. Scientists have assured consumers of the safety of this type of crops, but food and environmental activities discourage many countries to consume the crops, claiming they are unnatural and therefore unsafe. Now let us watch this simple video clip that talks about the issues and concern of genetically modified organisms. And as you watch, please take down notes of the important information, and you will be graded 10 points additional credit for your class standing.
GMOs are one of the most controversial areas of science. Genetic engineering is used in many fields, but even though medical applications like GM insulin are widely accepted, the debate heats up when it comes to food and agriculture. Why is that? Why is the same thing treated so differently? Let's try to get to the bottom of this and explore the facts, the fears, and the future of GMOs. Humans have been genetically modifying plants and animals for thousands of years. Maybe a few of your crops had very good yields, maybe one of your wolves was especially loyal. So you did the smart thing and bred the plants and animals that had traits beneficial to you. Traits are just an expression of genes. So with each generation, those genes got more pronounced. After thousands of years, almost every single plant and animal around us is vastly different from its pre-domesticated state. If humans have been changing genes for millennia, what makes a so-called genetically modified organism or GMO different? Selective breeding is basically hoping for lucky hits. Genetic engineering eliminates this factor. We can choose the traits we want, make fruit grow bigger, immune to pests, and so on. So why are people concerned about them? Let's start with one of the most common objections to GMOs. Gene flow, meaning GM crops could mix with traditional crops and introduce unwanted new characteristics into them. There is a method that might guarantee complete prevention, but is a big anti-GMO argument by itself: terminator seeds. The idea is that they could produce sterile plants, requiring farmers to buy new seeds every year. The very concept of this, however, caused a public outcry, stopping the technology being put to use. This brings us back to the unintentional spreading of engineered DNA. There have been cases of GMOs growing where they weren't planted and traces of modified genes found in foreign crops. But GM plants can't run wild entirely. Many crops pollinate themselves, and all crops have to be related to mingle. There are also cultural methods like buffer zones to keep unintentional crossing at a minimum. But if it's possible in principle that a GMO could unintentionally cross with a non-GMO, there's actually a more important question. Is food that comes from GM crops different to food from non-GM crops? This question has been a major concern from the very beginning. GM plants that are destined to be eaten are checked for possible dangers, and the results are evaluated by multiple agencies. After more than 30 years and thousands of studies, the science is in. Eating GMO plants is no more risky than their non-GMO equivalent. But don't just take our word for it. Sources for this and other claims are in the video description. But what about plants that have been engineered to be toxic? For example, Bt crops. A gene borrowed from the bacterium Bacillus thuringiensis lets engineered plants produce a protein that destroys the digestive system of specific insect pests. The plant makes its own pesticide. Insects that eat it die. That sounds alarming. Pesticide sprays can be washed off, while the poison in Bt crops is inside the plant. But actually, it's not a big deal. Poison is really just a question of different perspectives. What's harmless to one species might kill another. Coffee, for example, is a poison that kills insects but is harmless to us. Or take chocolate. It's dangerous for dogs, but a pleasure for humans. Bt crops produce a protein that is tailored to the specific design of the digestive tract of certain insects. It's completely harmless for us. There's also the opposite approach: plants that are engineered to be resistant to certain weed killers. This way, farmers can use them widely, killing the other plants competing for resources without harming the crop. Here we get to the dark underbelly of GMOs. For the pesticide industry, they are big business. Over 90% of all cash crops in the US are herbicide resistant, mostly to glyphosate. As a result, the use of glyphosate has increased greatly. That isn't only bad. Glyphosate is much less harmful to humans than many other herbicides. Still, this means farmers have a strong incentive to rely on this one method only, casting more balanced ways of managing weeds aside. That's one of the most fundamental problems with the GMO debate. Much of the criticism of this technology is actually criticism of modern agriculture and the business practice of the huge corporations that control our food supply. This criticism is not only valid, it's also important. We need to change agriculture to a more sustainable model. GMOs as a technology are actually an ally and not an enemy in that fight, helping to save and protect nature and minimize our impact on the environment. Let's look at some positive examples. Eggplant is an important crop in Bangladesh, but often whole harvests are destroyed by pests. Farmers had to rely heavily on pesticides. Not only was this very expensive, farmers also frequently got sick. The introduction of a new GM eggplant in 2013 stopped this. The same Bt protein we talked about before, an effective killer of insects but harmless to humans, was engineered into them. This reduced insecticide use on eggplants by more than 80%. The health of farmers improved and their income rose dramatically. And sometimes, a GM approach is the only option. In the 1990s, the papaya industry in Hawaii was under attack from the ring spot virus, which threatened to wipe out Hawaiian papaya. The solution was a papaya genetically modified to be vaccinated against the virus. Without it, the state's papaya industry would have collapsed. All these stories show a very narrow application. 99% of all GMOs we use right now produce pesticides or are resistant against them. There is so much more we could do. Scientists are working on GMOs that could improve our diet. Plants that produce more or different nutrients, like fruit with higher antioxidant levels that help to fight diseases, or rice with additional vitamins. On a larger scale, we're trying to engineer plants that are more resilient to climate change. Plants that can better adapt to erratic weather and adverse soil conditions, making them resistant to droughts or floods. GMOs could also not only reduce agriculture's impact on the environment, but actively help to protect it. Scientists are working on crops that can draw nitrogen from the air like microbes. Nitrogen is a common fertilizer, but its buildup pollutes the groundwater and speeds up climate change. Plants that collect their own nitrogen could fix two problems at once: the overuse of fertilizers in the developed world, as well as the shortage of it in developing countries. We could even modify plants to become super effective carbon collectors like the American chestnut tree, to mitigate and actually reverse climate change. With the tools we have today, our imagination is the limit. The world eats 11 million pounds of food every day. A UN estimate suggests we'll need 70% more by 2050. We could grow that food by clearing more and more forests to create fields and pastures, and by using more pesticides. Or we find a way to do it on the land we've got right now with more effective methods like GM crops. Intensifying farming instead of expanding it means GMOs could become the new organic. In a nutshell, GMOs have the potential to not only drastically change agriculture, but to also dampen the effects of our own irresponsible behavior. GMOs could be our most powerful weapon to save our biosphere. This video took more than 600 hours to make, which would be impossible without viewer support on patreon.com. If you'd like to support carefully researched content made with love, it's really very helpful, and you can get your own bird as a reward. If you want to learn more about genetic modification, we have more videos explaining the opportunities and risks of the technology and how it could impact our future.
Now we are down to the issues and concerns of genetically modified organisms. Genetically modified organisms are relatively new, and like any other new, there are contradictory perspectives about several issues regarding the utilization of these organisms. Some of these includes the following. First, if GMOs are safe to be consumed. One area that appeals to the attention of many is whether these genetically modified crops and fishes are safe to eat. There is no data to indicate that consumption of genetically modified organisms is bad for human health. GMOs have gone through more throughout tests than any other group of plants that people actually consume. In some aspects, GMOs vary from a type or typical plant by the inclusion of just one or two genes, which creates one or two new protein. The source and function of this protein are clearly understood. For instance, the protein are tested to ensure they will not have any characteristics that may cause allergic reaction. According to the National Academy for Science, the United States first and foremost authority on the independent and unbiased information on issues of science and technology. GMOs are harmless for human health. A great number of prominent health and science organizations from around the world have arrived at the same conclusion. Moreover, during the past 2 decades that GMOs have opened the consumers, there have been no incidents of health problems caused to genetically modified organisms. As of the present, GMOs have no health benefits to ingesting them over a non-GMO foods. Yet, this may change in the future as technology develops and becomes more sophisticated. Second is if they pose environmental threats by introducing them in underdeveloped countries. There are comprehensions that the introduction of genetically modified crops could have caused the decline in biodiversity, predominantly in regions wherein a crop originated and an extensive range of natural genetic variation is found. There might also be unforeseen supercussion of gene transfer, also termed as gene flow, among plants. An example is that there could possibly be an intermediable or uncontainable escape of gene in nearby wild plants via pollen. Concerns of the pest or weeds that might obtain resistance to crops have also been raised. In this case, the danger of gene flow is essentially assessed on the case-to-case basis. Gene flow happens extensively throughout nature. Its consequences is the primary determinant whether or not it is acceptable. The potential risk may depend principally on the specific crops and characteristic. People are not convinced and potential undesirable outcome of gene flow in certain areas are enough to dismiss the propagation of genetically modified crops away from developing countries. Also, there are several ways of inhibiting and regulating gene flow. It is imperative to have adequate seed banks to preserve genetic resources of crops effectively. Third, is GMO are classified as artificial crops? Numerous people are alarmed that genetic modification is artificial. Debates regarding being natural are complicated and evaluate many intricate issues. The transmission of genes amongst species is frequently believed to be mainly objectable since it disrupts boundaries between natural species. All forms of plants breeding have altered specific crops or biodiversity in general, both in direct or direct ways. Hazards and benefits of particular inventions need to be considered in single cases. The simple arguments about artificiality is not convinced enough to stop the responsible exploration of the potential of GMOs. People have been producing foods that have undergone processes in the laboratory to improve the palatability and general appearance to look more appetizing, and all of these are synthetic methods performed on food as well. And lastly, if biotechnology corporations are efficient in controlling most of the technique or technology needed to develop GMOs, several agriculture biotechnology corporations regulate most of the technology required to improve genetically modified crops on top of the agri-chemicals and crop germplasm. Several are also alarmed that the corporations and those who possess intellectual property rights have unjustified influence over the accessibility of genetically modified crops. Access to this technology and germplasm is critical for supplementary researches. Furthermore, much of the present genetically modified studies are only limited to the advantages of large-scale or large-scale farmers in industrialized nations, such as concentrating on traits of crops regarding herbicides tolerance. Additional resources should be committed by government worldwide to finance a major development of genetically modified related research significant to the needs of small-scale farmers in unindustrialized countries. In conclusion, genetically modified organisms are alive. They have the ability to migrate and proliferate globally. Incidentally, clear signals ought to be delivered to biotechnology laboratories to proceed with added caution and evade instigating unintended damage to public human health, to biodiversity, and the environment. While it is widely assumed that consumers have the right to demand mandatory labeling of GMO food products, self-regulating evaluation for safety and environmental effects, and accountability for any harm linked with GMOs, international leaders must look into these concerns of this technology-driven society. Many regulatory laws already exist for risk assessment which are performed on the three levels of impact on agriculture, such as gene flow, reducing biodiversity, food and food safety, such as allergenicity and toxicity, and environmental with an on-target organism, and this make the public reassured of their safety on the issues and concerns of genetically modified products. In addition, recent the Cartagena Protocol has formulated policies and guidelines and has compelled countries and corporations to conform by when the product, handling, and consumption of genetically modified materials. It is therefore up to the public judgment whether to get involved or not in the technology of genetically modified organisms. For as long as there are risks in the consumption of these genetically modified products and are not properly addressed, it is certain that the present society will continuously follow the liberations of genetically modified organisms and the related regulatory issues in the years to come. Now, this is the end of our discussion, but let me leave you with this quotation. We must address individually and collectively moral and ethical issues raised by cutting-edge research in artificial intelligence and biotechnology, which will enable significant life extensions, designer babies, and memories extraction, according to Klaus Schwab. And these are the references that I made use for this topic. Thank you for learning with me today, but please don't forget to click the like button and subscribe. See you on my next discussion. Goodbye!